വിചാരിച്ചിട്ടാണ്. എല്ലാവർക്കും, those who are absent, you can go through the videos which we are putting in the Moodle. Okay. So today we are going to study about the histology of placenta and the umbilical cord. Okay. We have studied the embryology of placenta. We have studied the embryology of umbilical cord. So umbilical cord, how we can see the umbilical cord? This is the structure, how it appears in a section. This is a structure showing the umbilical cord. Being a vessel, it can be seen somewhat rounded in nature. Okay, so the umbilical cord, we have studied umbilical cord that is forming within the vitelo-intestinal duct or the connecting stalk, within the connecting stalk when it is attaching. When we have discussed about the folding of embryo, we have seen the secondary embryonic sac which is continuous into the connecting stalk towards the attachment of placenta. Later it will convert into, after the formation of embryonic fold, isn't it? Head and tail fold. After the formation of foldings of the embryo, that will convert into a small narrow channel which is connecting in between the fetal and maternal parts. So the major transmission occurs in between the fetal and maternal blood through means of this umbilical veins and arteries. Understood? So the umbilical arteries and veins, they can be paired. These umbilical arteries and veins can be seen as paired, and in later development, one of the umbilical vein disappears. Only left is left, or one. There will be a right and left arteries, right and left veins. These right and left arteries remains intact, while the right vein will disappear and the left will maintain. So that in a complete development, during the later stages of development, we can see the embryonic umbilical cord with two arteries with single vein. Can you see like this? This is the diagram showing the cut section of an umbilical, histological section of an umbilical cord. This shows the major important features. It is very easy slide to identify. There are three openings can be seen majorly, and there may be another additional opening that represents the remnant of vitelo-intestinal duct or allantoic duct can be seen. There may be the presence of the remnant, or else it can be seen like this. This is our college slide where we can see two umbilical arteries and umbilical vein. This is a slide that we have to study in our department. This is umbilical vein. We can see the umbilical vein and the two arteries. Umbilical vein is more collapsed. The lumen is larger and more collapsed. The arteries shows the blood stains within it, and these are the walls of the arteries where we can see the smooth muscle bundles, and the space which is presenting in between, or this rest of the area, that represents the Wharton's jelly. The Wharton's jelly is developing from the mesenchyme, isn't it? So that we have studied in the embryology. So the Wharton's jelly is the material that is can be seen between the arteries and the veins. So that is the substance which separates the arteries and veins can be seen in the umbilical cord. So this is the diagram, this is the appearance of a cut section of umbilical cord under the microscope. This is a low magnified view. This is a magnification of 5. That is object I have used is the 5. That is, the speciality is that when it is when we are using higher magnifications, we can't see this much of vessels in a single view. That's why I made it for, usually we will study all the slides under 10x, but here I have used 5x because we want to see all these features in a single view. That's why. Okay, so these are umbilical arteries and veins. These are the major features that can be seen in a cut section of umbilical cord, and it is very easy to identify. This is the unique features that we can see, like two eyes with a mouth, isn't it? It's somewhat like that. Can you feel like that? So that is the speciality of the umbilical cord. The structure consists of mesodermal connective tissue, that is the Wharton's jelly, and it is covered with amnion, that we know. Then it contains an umbilical vein which is connecting the placenta to fetus. Placenta to fetus, so that it contains oxygenated blood. Two umbilical arteries are there. Two umbilical arteries are there, fetus to placenta, so that it is containing deoxygenated blood. See, usually the arteries are carrying oxygenated blood and veins are carrying deoxygenated blood, isn't it? But in case of umbilical cord, it is ulta, that is the arteries containing deoxygenated blood and the veins containing oxygenated blood. And there we can see the remnants of yolk sac and allantois also can be seen. There is a probability. So in this diagram it shows the vasa vasorum. Vasa vasorum means the blood vessels supplying the vessels, that is named as a vasa vasorum. So this arteries and veins, they are supplied by the small arterial vessels, that is representing here, that is vasa vasorum. And we can also, there may be the presence of allantoic duct or vitellointestinal duct can be seen there, that this is the remnant of vitellointestinal duct. Umbilical veins you can see, and the connective tissue, mesenchymal connective tissue, the Wharton's jelly. To umbilical arteries we can see, to umbilical veins. This is the early stages of development. Later it will convert into only three structures. Vein, one of the vein will leave. So this is the diagram that we have to draw in the histology rough record and fair record. We can draw two umbilical arteries with one umbilical vein. The shape, external shape, that depends. It have to be somewhat like rounded. No need to make these depressions and projections. That's up to you. You can make an outer rough rounded structure with two umbilical arteries with one umbilical vein and allantoic duct, or the vitellointestinal duct you can draw, and with the pink shade you can draw the Wharton's jelly also. And this is outermost layer is the amniotic epithelium. So that is the speciality of umbilical cord. Have you understood the umbilical cord? It is very simple to identify, isn't it? Is this slide is identifiable, or it is easily we can identify, isn't it?
Yes, ma'am.
Okay. It is unique features. These features only can be seen in the umbilical cord, so that it is very easy to identify under microscope. In terms of a number of, when we are discussing about a number of slides, it is very easy to identify. Okay. The next slide is a placenta. In placenta, we can see the cut sections of chorionic villi. These are the cut sections of chorionic villi. Okay, at different stages, you can see this each structures that represents the chorionic villi. The gap between that white areas which can be seen in between the sections, that is representing the intervillous spaces. That intervillous spaces are filled with blood stains, we can see here. That blood stains containing the erythrocyte cells, the maternal blood cells, can be seen in the intervillous spaces, and these are the structures which can be seen. This is the chorionic villi. You can see the outer layer that is formed with the cytotrophoblast and syncytiotropoblast, and inner this substance representing the mesenchyme and mesenchymal cells and or extra embryonic mesodermal cells can be seen, and these small openings that can be seen inside, that is representing the blood vessels inside. Okay, so it can be in a single section. This is the diagram showing a single chorionic villi. So each of this structure, if we are magnifying, this is the view that can be seen. So in a single section of chorionic villi, you can see darkly staining outer syncytial trophoblast. Darkly staining outer syncytial trophoblast you can see, and a single layer of lightly staining cytotrophoblast inside also you can see. Can you see the two layered cells here?
Yes, ma'am.
This is outer layer, the syncytial trophoblast, and inner layer the cytotrophoblast. Inside that, this area that is representing the extra embryonic mesoderm. And inside that we can see the fetal blood vessels. And inside the fetal blood vessels, in this diagram it represents the fetal erythrocytes. Majorly within this villi we can see two types of cells. That is, one mesenchymal cells, the other one is macrophagic cells. Two types of cells can be seen in between the fetal blood vessels and the trophoblast cells, that is in the extra embryonic mesodermal region. There we can see two types of cells. One, fusiformal mesenchymal cells, that is differently shaped mesenchymal cells can be seen. Along with that, there is a presence of macrophagic cells which have got a phagocytic action. That macrophagic cells are named as Hopf cells. Macrophage cells and the different types of mesenchymal cells also can be seen at this region. Okay. Then the fetal blood vessels, that is can be seen at the core of each villi. At the core of each villi we can see there is a fetal blood vessels. Inside the fetal blood vessels there are the erythrocytes. Erythrocytes are the blood cells, isn't it? So erythrocytes, fetal erythrocytes can be seen inside. Okay. And this is somewhat like a full grown or tertiary villi, and here it is starting only. So we can see different stages of villi can be seen, because the villi can be seen in a tree like appearance, isn't it? Different branching patterns are there, so that in a section we can see different stages of sorry, different stages of developing villi can be seen. And this is the intervillar spaces between the villi. They are intervillar spaces. These intervillar spaces containing maternal erythrocyte cells. It is containing maternal blood, so that these red structures, that is representing the maternal blood cells. Understood?
Yes, ma'am.
Okay, so all these are the features I think already we have studied, isn't it, in the embryology, so that it will be familiar to you, isn't it? So it can easily identify how it is appearing. So this is a diagram showing the different stages of development with outer trophoblast, syncytial trophoblast and cytotrophoblast, and inside we can see the blood capillaries and the space between, which is separating the fetal blood capillaries from the trophoblast. That space is representing the extra embryonic mesenchyme or the mesodermal part. Understood?
Yes, ma'am.
Okay, so that is all about the two slides, placenta and this one is umbilical cord. Okay, placental features, that is the cut sections of chorionic villi present that we have seen. The trophoblast cells are visible as an outer dark staining syncytial trophoblast and inner outer dark staining syncytial trophoblast, inner light staining cytotrophoblast. Like this, outer dark staining syncytial trophoblast, inner light staining cytotrophoblast. The core of chorionic villi shows the mesenchyme or embryonic connective tissue with two cell types, mesenchymal cells and the macrophages. The presence of blood vessels are located at the core of the villi. The branches of umbilical vessels can be the veins and the arteries, isn't it? The branches of umbilical veins and the arteries can be seen inside. The intervillous spaces are with maternal blood cells. The intervillous spaces are filled with maternal blood. Have you understood?
Yes, ma'am.
Okay. So you can draw the diagram now itself. This is the diagram that we have to draw for the umbilical cord, and this is the diagram that we have to draw for the placenta, showing the maximum features. Okay.
Okay, ma'am.
Okay, draw it and submit it before 12 o'clock. Okay. It is very easy to draw. I will send it in WhatsApp now itself. Okay.
Okay.